anybody who might be watching, I guess. Um, really excited to talk to you about some of the work that I've been doing. I'm a postdoctoral fellow in Tanya Corteme's lab at the University of California, San Francisco. And we're exploring ways to use deep learning to explore sequence function landscapes. And so, yeah, thank you for the opportunity to share my work. And if you have any questions, feel free to interrupt just as I go along in the presentation. So as the title mentions, I'm really interested in these proteins called protein tyrosine kinases. These are really important in biology in general. So here I'm showing you an example of a protein tyrosine kinase called HER2. HER2 is a really important receptor tyrosine kinase. You can see that it has three domains. This extracellular domain, which is on the surface of a cell, this transmembrane domain, which is within the membrane, and then this kinase domain, which does a lot of the important signal processing that happens in the cell. Normally, this functions to do things, maintain normal cellular functions such as cellular proliferation, cell growth, these types of things. But if you have small mutations in the kinase domain, so here I'm showing you, a small mutation being this red streak in the kinase domain, a small mutation can completely change the activity of this kinase domain and cause it to, have phenotypes that cause things like oncogenesis or cancer or things like that. So understanding how these types of mutations affect the functions of these kinase domains is really important, not only from a basic science standpoint of understanding how these proteins work, but also for human health in general. And so here I'm talking about HER2. It's actually been found that you can build drugs to this as well in order to stop cancer. So it's a really important target to learn about. And so the idea here is that, we'd really like to know what the effect of these mutations are in these kinases, because they have really, really important implications for, human health. And it would also just be really important to know for basic sciences of how these kinases work. So if you can, map the functional effect of any mutation and say, oh, this mutation causes the kinase to activate or it causes it to lose function, it would be really helpful in order to predict the effects of any mutation in a kinase. And so, if we're talking about learning about these types of effects from these mutations, what we're really talking about is a sequence function landscape. And being able to map out any mutation to, any function in the kinase would be a really important advance for biology. So like we were just talking about, if you have a mutation that activates a kinase, it might be really useful in learning about, some disease that, such as cancer. There might be mutations that affect the allostery in a protein. So, in response to this signal, it has this function, and that would help improve our understanding of just protein biology. And I think in a more engineering standpoint, if we learn about, the specificity of kinases, it might be useful for designing new types of orthogonal signaling pathways. But, this just stresses the importance of learning about, what do these mutations do, and so that we can understand these proteins better. Exploring that sequence function landscape, however, is really, really challenging. So here I'm showing you kind of a cartoon of the sequence landscape of, in our case, a protein kinase, but really it could be any protein. And the challenge here in exploring this sequence landscape is that sequence landscape is massive. It's also, sparse, which means that function is very rare within the sequence landscape. And it's also very rugged. And so what I mean by each of those is that, for any 100 amino acid protein, you have 20 to the 100 sequences that could encode, that you have to explore. So it's a huge and massive sequence space to explore. Within that massive sequence space, only a couple of sequences really are functional. And so, the ability to explore non-zero activity proteins is very challenging. And then ruggedness is talking about, you can make one mutation to a kinase that completely kills its activity. And so because of these reasons, it's very challenging to be able to explore the sequence function landscape in any efficient way. And so what we propose is that we can use deep learning in order to
Yeah,
do
you can
this.
change it.
Excuse me?
Sorry, I think it was background noise, but we can continue.
Oh, sorry. Were you not hearing or seeing my screen?
No, someone I think in the background they
Oh, I
talked,
see.
but we can't get it.
Gotcha.
Yes.
Gotcha. Okay, no problem.
Mhm.
Um, so yeah, so in order to explore the sequence landscape, we propose that we can use deep learning in order to do this. And this takes advantage of many of the advances that deep learning has provided us with, especially in the protein space. So specifically what I'm talking about is our deep learning models that are able to basically go between sequence and structure. And so here I'm just showing you a diagram. We can go from the forward direction. If we go from a sequence to a structure, this is what AlphaFold does.
Doctor,
Yes.
Dr. Kosuke, previously I think the previous slide, you've talked about the landscape in terms of the function landscape, and also
Mhm.
talked about the sequence landscape. So my question is about this work is focusing on the function or sequence, or it is about the mutational landscape of for example, tyrosine being uh, being sort of like sparse, but also like it is wide in a way that you have a number of possibilities. So the question is about the landscape we are focusing on here. Is it mutational or functional or sequence itself?
So I think it's a little bit of both. In my work, we explore the sequence space and then measure the functions of many sequences. Does that answer your question?
Okay, yes, I think it clarifies. Yes,
Okay, yeah.
yes.
Um, okay, great. Um, So we were talking about how sequence landscape is very sparse, and in one direction, going from sequence to structure are things like AlphaFold. But what's also really exciting is now that we can take a protein structure and we can go in the reverse direction. So these are models that you might have heard of, the most prominent being something called protein MPNN that came out of the Baker lab. And so what you can do now is just give these models a structure of a protein kinase that you might be interested in, and then it can propose, sequences that can fold into that kinase sequence. And so going back to this kind of figure, really the specific question that we're interested in is, can we use these types of inverse folding, so going from structure to sequence, in order to access, new parts of sequence space, and then can we measure those, in order to be able to really understand what the sequence function landscape looks like for, a given protein kinase. Um, so the first question then is, can you even do that at all, right? Um, So what we did is I worked with one of my lab mates, Denise, who developed one of these programs called Frame to Seq, which is also goes from structure to sequence. And so what we did was we first focused on a receptor tyrosine, the kinase domain of a receptor tyrosine kinase called EPHB1. This is a really important tyrosine kinase that facilitates cell-to-cell signaling and cell-to-cell communication. And the structure of this protein has already been solved. And so we can take the structure of this protein and we can use these types of models in order to design sequences that we can test. The approach to do this was published in a recent paper from David Baker's lab. And so the general approach in order to do this is first, you take the structure and then you at first give it a set of constraints. And the set of constraints is that you need to keep important residues that are within the proteins. And this is just because, like we were talking about, that sequence landscape is really rugged. So you need to at least, and sometimes some of these residues are really important for function and that you can't mutate those. And so we find those important residues firstly by looking at like the functional regions in this protein. And so for example, in this protein kinase, there's the ATP binding site, which is really important, and also the peptide binding site. These are basically sites in the protein that are important for binding the substrate of the protein, which is ATP and a peptide. And so those are structural regions where we say to the model that you can't mutate these residues. And then the second class of residues that we fix is residues that are conserved in evolution. And so what we do is we look at the multiple sequence alignment of this EPHB1 protein, and we find residues that are basically conserved throughout evolution. So here I'm showing you a graph of position on the x-axis and conservation on the y-axis. And you can see that, for example, there are many residues that are 100% conserved in all protein tyrosine kinases. And so we set a couple of thresholds, which I'll talk about later, and we say to the model that we can't mutate these residues. So once we do that, we pass it through these redesign models. So we use both protein MPNN and frame-to-seq. These are both deep learning models that accomplish the structure to sequence task. And then we use, we design many sequences to do this. And then we filter the best ones by using ESM1V, which is a protein language model. And so we use this just because some of those sequences might not work or be predicted to be functional. And so we use ESM as kind of a filter just to get the best 20 sequences or so. And so we do this in order to, just test the hypothesis that we can use these structure condition sequence design models to redesign these kinases. And so really quickly, I'll talk about how we verify that these are active. So this is more of an experimental workflow, and I'll try to be brief because I know this is more of a computationally focused group. We use something called a cell-free expression system, which is basically a way to make proteins outside of a living cell. And so what we do is you encode the DNA of the protein in a gene fragment, and you provide it with the transcription and translation machinery that you need in order to make a protein. And so we can express these proteins very quickly in a test tube. Here I'm showing you a gel, an SDS page gel, just showing that we can express these proteins when you provide it with chaperones. And then the important part is this graph on the right axis, on the right of your screen. And so this graph is just showing you, on the x-axis is time, and on the y-axis is fluorescence. And we can basically measure the activity of a kinase by looking at its activity on a substrate, which results in fluorescence. So here I'm just showing you that in the presence of the wild type protein that we're interested in, EPHB1, you can see that this enzyme, acts on its substrate over time. And in the absence of this enzyme, it doesn't work. And so this is just a really nice way in order to quickly see, which sequences work. And so using that previous method, we designed about 28 different proteins, and then we tested them using this experimental workflow to see which ones would work. And so this is that data. I'll go into, there's a lot of data here, so I'll go into the specific ones of interest in a little bit more detail, but here I just want to show that we can use these types of experimental workflows in order to really quickly see, if we can actually get to sequences that work when you redesign these kinases. And so here, I'm just showing you the controls of the experiment. So you can see in WT, that's the wild type enzyme. Here you can see that the wild type works as you would expect. And when you don't have a kinase, you don't have any activity. So we're looking at the green lines here. And what's really nice is that when we test these kinases, you can find some that are active. And so we talked about, that when we want to fix residues that are evolutionarily conserved, and it turns out that fixing residues that are, at least in 50% of all protein tyrosine kinases gives you a pretty high success rate. And so you can see that these designs are active in the presence of the substrate. And what's really nice is that these designs have, over 80 mutations from the wild type enzyme. And this is all came straight through that deep learning model. And so we find that we can indeed use these types of models in order to design new sequences, that nature has never designed before. And just to show you some failed cases, here are some examples of if you give the model basically, the freedom to redesign almost all residues except for only the most conserved residues, you can see that in general, these protein designs don't really work very well. And so we were able to figure out, how to use these models in order to give us sequences that are active. And so this answers that first question, which is, how do we use these actual deep learning models in order to design sequences that might be functional? And indeed, we are able to use them and design sequences that are far away from the natural sequence, but still remain functional. So we can explore the sequence function landscape in a much greater way than we could have before. And so since we now know that we can do this in a relatively successful way, we then wanted to do this at scale. So we told you that exploring the sequence function landscape is massive. And so we wanted to not test 20, but we wanted to test hundreds or even thousands of these sequences. And so what we did was we turned again to the deep learning model and we designed more sequences. One caveat is that due to the experimental method, it's actually difficult to synthesize the full length gene. So what we did was we took that human EPHB1 kinase that we were interested in, and we only focused on one specific region, which we called the redesign region, which is highlighted here in blue. And in blue here, even though it's only part of the kinase, it actually encompasses many of the really important functional regions of the kinase. So it has, elements of the ATP binding site that we were talking about before. It has elements of the peptide binding site that we were talking about before, but also has other structural features, such as like the activation loop or the spines of the kinase. And so we're only now redesigning a portion of the EPHB1 kinase. And then we basically go through the same workflow as before. We identify the residues that are fixed within these. We identify the residues that are structurally important. We identify the residues that are evolutionarily important. And then we go through the same protein MPNN and frame-to-seq redesign protocol and filter by ESM1V. And we end up with about 2,000 designs. And our goal really is to test many of these so that we can explore the sequence function landscapes of these enzymes in a, relatively broad way. And so what do these sequences look like? So after doing all of this redesign, in all 2,000 of these sequences, we see mutations at about
Doctor.
76. Yes.
Yes, Doctor, there is a question. Someone is asking, how do the models ensure that the fixed residues, like those in the ATP binding site, remain unchanged with the rest of the sequence being redesigned? Also, is there
while
specific...
the rest of the sequence is being redesigned, is there
Yes.
any specific constraint or loss function built into the model to handle that?
Yeah, so let me talk. So the first question is, how do you ensure that the
Fixed residues.
specific residues aren't redesigned? And that's a parameter that you can explicitly you can be explicit about when you do the redesign. So you in the model, there's a parameter that you can say, fix residue 24. And so when you do that, it'll basically not redesign that 24th residue. And you could do that for many, many residues. And so that's how you ensure that those are not redesigned. Can you repeat the second question?
Yes, the question was written, I had posted it in the chat. Is there a
Mhm.
specific constraint or loss function built into the model to handle that?
Is there a specific like function built into the model?
Yeah, to handle about the rest of the sequence being redesigned.
Yeah, so I think, yeah, so the model does have the, like we were talking about, does have that specific parameter in order to tell it which residues you can and cannot redesign. Um, or were you talking about like function as in like enzyme function, for example?
I
No,
think
like,
I think he's talking about. Okay, go ahead, Herbert.
look, man, what were you saying?
I think the question was about the constraint, or which I think he has addressed the parameter that handles some specific sort of design interests within the protein structure.
Yes, in his first explanation, while he was answering the first part, he answered the second part too, because he was trying to explain. Yeah.
I think, Doctor, you've answered it, but also for me, something
Mhm.
close to that is about the somewhere I saw that there is an effect from the length of the residues on the functional sort of outcome of the redesigned sequencing. So
Mhm.
and it looks like longer residues tend to produce sequences that have functions.
Mhm. Um,
Yes.
when you say longer residues, do you mean like more residues that are redesigned or something else?
I mean, like depending on the shorter residues, but also long. I'm talking about length, like the effect of length on the residues, and
Mhm.
the kind of functional outcome of of the mutation that might happen or the redesign. Yes.
Yeah, that's a really good question. So like, so the types of mutations that the models are incorporating are definitely really important. Oftentimes, the model will substitute similar residues. So I don't, if you're talking about like length, let's say like a tyrosine, a common mutation that you see, for example, is like a tyrosine to a phenylalanine, which is a very similar types of mutations in terms of length of the residue and things like that. And so the model does have some biases in terms of like the specific lengths of the residues, if that gets at your question.
Yeah. Yeah. Sure.
Yeah. All right. Okay. So yeah. So and so when we redesigned a lot of these enzymes, here you see what that sequence landscape looks like. We find mutations throughout this redesigned region, so at about 76 different positions in this region that we redesigned. And then if we look at the number of mutations we see that compared to the wild type enzyme, frame to seq is incorporating more than 30 mutations in every sequence, on average, and protein MPNN is doing a little bit better at incorporating about 40 plus mutations in every single enzyme. So we're really diverging from the wild type enzyme in a very big way. And so here you can see that protein MPNN, incorporates more mutations, but within those sequences, if you just look at how different are sequences within protein MPNN or within frame to seq, for example, we can see that, within frame to seq sequences, each mutation is about 25 mutations. Each sequence is about 25 mutations away, similar as protein MPNN. But if you compare, a frame to seq sequence with a protein MPNN sequence, you can see that those also have many, many mutations away from each other. And so all I'm trying to say here is that, like not only are we making many, many mutations, but also the use of these two different models is able to give us a broader range of sequences than if we were to only use, for example, protein MPNN, which is, kind of the state of the art that people typically use. And just to expand on some of these differences a little bit more, here we're looking at on the x-axis, frame-to-seq versus protein MPNN. And if we look at the BLOSSOM score, which is kind of a measure of how natural a mutation is, you can see that the frame-to-seq mutations are generally, slightly negative, which means that the mutations are less conservative than you would find typically in nature. And if you just look at the sequences and do a principal component analysis, you can see that frame-to-seq sequences and protein MPNN sequences are very distinct from each other. And so again, this is just showing that frame-to-seq and protein MPNN explore very different sequence spaces and design unique sequence solutions to the problem of, given a structure, design a sequence that folds into that structure. And so the use of both models is important for our work. With all these sequences, it's time to just measure all of these sequences. And so again, we're using this similar cell-free expression workflow that we used before, except we're doing it at scale. So we're using things like 384 well plates in order to measure these in high throughput. The other thing that we do is not only do we measure activities. Oh, yes.
Doctors, on the previous slide, I'm seeing something interesting. Yes, on the right graph, this distortion, not distortion, but the alignment where you have the gray sort of like brown on that is like, what is that signifying? Like, because when you look at the first one, the alignment is straight. You can see like the MPNN is performing sort of like on the BLOSSOM, giving a higher score. But on this one, on the right, this alignment is, does it, what can someone pick from it? Like, it's a little bit fascinating, sort of like aligning in that sort of form. So is there anything someone can take away from this?
Yeah, I think the big takeaway message is just that the models are different and they design different sequences. And we did do a little bit of analysis in the paper of what exactly those differences were. And typically it's about the specific mutational preferences of each model is slightly different. So for example, sometimes, frame-to-seq might prefer to substitute, let's say, a leucine with an alanine, for example, while something like protein MPNN might decide that, an alanine to a tryptophan or something, a mutation might be more preferable. So it just has slight different biases, and that I think has to do with they're actually trained slightly differently and on slightly different data sets. And so they might have learned different things during that process.
Perfect. Thank you.
Yeah, of course. Yes. And so, when we do these measurements, we not only now measure activity like we did before, but we also measure expression level through this assay called split luciferase complementation. And I don't want to get too much into the details, but what this does is that we can now measure the concentration of the enzyme within each reaction. And so what we can do is we can measure the activity per enzyme. And so that gives us a specific activity, which gives us kind of the intrinsic activity level of each enzyme. And so because we do this in cell-free and we're using these 384 well plates, here I'm just showing you, it's not super important what each graph here is, but we can measure, each graph here is one enzyme variant, and we can measure lots of these enzyme variants within a single experiment. And so we can generate a lot of data, which is, basically very useful because we would eventually like to use some sort of machine learning model in order to learn about these enzyme variants. And so at the end of the day here, what we collect data on about 537 different kinase variants. And here I'm just going to show you a bunch of histograms. And so here on the x-axis is that specific activity level of each enzyme. And then on the y-axis is counts. And in blue here is protein MPNN, and brown is frame to seq. And so you can see that we see a very wide distribution of activity in both protein MPNN and in frame to seq variants. Here in the next graph, I'm showing you a similar type of plot, but instead of measuring specific activity, we're measuring the expression level or the concentration of the enzyme. And so what's interesting, you can see here already, if you stratify this by the different models, you can see that, like we were talking about, the models are trained slightly differently and have slightly different mutational preferences. And that does end up in, difference in performance in some of these enzymes. So for example, the frame to seq enzymes generally are more active, but they are, on the whole, expressed at lower levels than protein and PNN designs, which are generally slightly less active, but have higher expression levels. And here I'm just showing you that the activity, that the experiments are very robust. So when you repeat these experiments, you get the same measurements back. And so we have a very high quality data set now of enzyme variants linked to their specific activities and to their concentrations. And so because we have all this data, we can do some interesting data. So we were talking about before, just now, like frame to seq variants generally tend to have higher specific activity levels but have lower concentrations. And if we look at that data a little bit more, we can see that there does appear to be some trade-off between activity and expression, which is interesting because it suggests that maybe the higher activity enzymes are maybe less stable than you'd expect, and that there might be an inherent trade-off between those two functions. We see the same type of trade-off in protein and PNN design. So you can see high activity variants are typically found at low concentrations, whereas variants that have very high concentration are typically low to moderate activity. So I think that's an interesting trade-off that suggests that maybe this is something inherent to many proteins, which is that as you get more stable, you get less active, for example. And so this was done in a very high throughput way. We collected, hundreds of data points all at once, but we also wanted to do a couple of checks just to make sure that this data was really robust. So we did, kind of the gold standard in biochemistry, which is to do Michaelis-Menten analysis. And we did this on about 13 of these variants. And what you can see here on the x-axis is the activity that we measure in our high throughput assays, and on the y-axis is the kcat over km. So the specific a measure of the specific activity of each enzyme, and this was done by like rigorously purifying the enzyme and doing all the Michaelis-Menten analysis on each of those enzymes, and you can see that in general, the activities that we measure in high throughput tend to match with the activities that we measure when we do much more detailed analysis. And so that suggests to us that our data is of high quality. From a biochemical perspective, what's also interesting is that most of the data we observe doesn't change with Km. So what that means is that these enzymes are different not because they bind to the peptide substrate better, but usually it has to do with the inherent catalytic activity of the enzyme kcat. So you can see that if we compare the correlation between kcat over km or just kcat alone, we see very, very similar performance. So what we're measuring is differences in like the catalytic performance of the enzyme. And so, this part, I just want to emphasize that we have a lot of data We have, hundreds of enzyme variants that we can look at. And we also have measurements that are very reliable. And so because we have that, we were curious if we could train a model to learn what's actually going on in these enzymes. And so we used a very simple, I hesitate to even say machine learning model, because what we're doing mostly is regression. And we're curious to see if we can use, simple models such as, ridge regression or linear regression in order to predict the activity of kinases. And so I did this in collaboration with another lab mate, Amy. What we did was we were interested in seeing three different variables within these types of models. So one, we were looking at protein embeddings. So these can be things like ESM embeddings from a protein language model, or it could just be, like a one-hot encoded sequence. There's been some work showing that if you provide more information, so here's what we're calling augmented parameters, you might be able to improve a model. And then we're looking at the types of models themselves. So here I'm just showing you all the different variables that we tested. We tested, on the embedding side, we tested ESM protein language model embeddings. We tested these Georgiev encodings, which have information about the physical and chemical properties of each amino acid. And then we just tested a one-hot encoding. On the augmented parameter side, we tested this evolutionary density from this POTS model called EV mutation. And we also used a predictor for the delta delta G of each mutation as an augmentation in our model building. And then we just tested three relatively lightweight and simple machine learning models. So we used ridge regression and lasso regression, and we also tried random forest. And so here are the results of these models. So on the x-axis here is all the models and different combinations of embeddings that we tried. And on the right, on the y-axis is the Spearman row correlation coefficient. And so what we can see here, interestingly, in terms of the model side, which is in different colors, you can see that ridge regression, just a simple ridge regression model does really, really well on these sequences and achieves an R of around 0.75 in the best cases. So the ridge regression appears to be the best way to model these protein sequence landscapes. We also, on the embedding side, we find that these ESM embeddings from the protein language model perform very, very well. And that is to be expected because these embeddings have a lot of information that's learned by the protein language model. And then we find that this augmentation actually doesn't provide much benefit. So you can augment the embeddings with evolutionary density or predicted delta G. But if you compare those augmentations compared to having no augmentation, these machine learning models typically perform just as well. And so what we concluded is that these ESM do really well on this task of learning the sequence function relationship. However, even though that's true, we, as those ESM embeddings are very difficult to interpret. And so what we actually ended up doing was building a model based on just the one hot encoding of the protein sequence, just because those are, easy to interpret. You can say, this position, this mutation at this position causes this effect. And so it's very easy to learn what's going on there. And so we built a model just on the one hot encodings. And so here I'm showing you just the general performance of that model that we built. You can see in Y is the x equals Y line. On the x-axis is the actual rates that we measured, and on the y-axis is the predicted rates. And you can see that there's a pretty strong correlation. And because we're using one hot encoded sequences, these parameters, this model is very easy to understand because the coefficients are very directly related to the mutations at each position. So if you just graph the regression coefficients onto each position, you can clearly see what's going on. So here I'm just showing you a heat map of like different amino acids on the x-axis and different positions on the y-axis. And you can see, for example, I don't know, I hope you can see my mouse. But at this 791T position, an X here represents the wild type, but you can see that the wild type threonine is very highly preferred, and mutations to this threonine are very detrimental. So for example, if you substitute that threonine with a leucine or a methionine, you get a really, really big hit in the activity. And so it's very easy to see what the model has learned. And because of that, it's really interesting to look at these types of mutations and see what the activity levels of these are. Especially with, for example, like something that's predicted very strongly and predicted strongly here, as in like predicted to be positive, or an activating mutation, and things that are negative, which are predicted to be, for example, like very detrimental. So for example, because we have this model, we can look very in depth at specific positions. So for example, in sequences that have this R6662E mutation, we find that this is a positive coefficient mutation. You can see that in sequences that have this, the median activity of those sequences is double that of the sequences without it. And so this mutation clearly has a very big impact on the performance or on the activity of a kinase. You can do the same thing with a negative mutation. So this K800D, if you have this mutation, generally sequences perform half as well as sequences that don't have that mutation. And so, because we trained on so much on hundreds of data points, we can really get to see specifically the effect of a single point mutation in these sequences, which I think is really, really interesting. If you look at all of these in general, if you have a positive coefficient, generally it's about a 5% increase in the ratio in the activity, whereas if you have a predicted negative mutation, I think it's about a 50% decrease in activity. And so the model, even though it's a very simple one-hot encoding based, ridge regression model, it learns like real effects from these mutations. We also find that the model only needs a couple of positions. And so here I'm just showing you, how many positions that we include in the model, and then the mean squared error on the y-axis. And you can see that the performance of the model actually saturates, at about 10 to 12 residues. And so what we interpret that to mean is that, there's only about 10 to 12 residues that are really, really important, even though we redesigned, 76. And so if you look at where those positions are in the crystal structure of the enzymes, you can see that a lot of these positions are very close to important functional regions of the kinase. So for example, it's very well understood that this alpha C helix, which is this helix right here, is very important to the function of protein tyrosine kinases. And you can see many of the residues that are important in the model are indeed like close to that alpha helix, which suggests that maybe those residues have some sort of important regulatory role in affecting the activity of the kinase. So I think it's really impressive that a really simple model can learn these types of relationships. And what we were also curious about is, okay, we have this model, can we predict the activity of an unseen variant? So I told you near the beginning of this presentation, we actually designed, about 2,000 sequences in our library, but only measured the activity of about 500. So what about the rest of those? What's the activity of those? So we predicted the activity of those variants using this simple model, and we categorized them into these bins where you have like the most active variants, high active, medium active, and then low or no activity variants. And then experimentally tested those. So these are variants that have never been seen by the model. And we can see that here, if we look at the experimental measurements of those graphs, they match up with the predictions quite well. So for example, the top 8 variants generally have higher activity than, the variance that we binned in these high activity measurements, which are higher than the medium activity variance. And then we find that the low dead variants are basically spot on. All these variants, don't work. And so this model is quite predictive of the variance that we see. And it can, sorry, I would say the model is, appears to learn important relationships in the sequence function landscape. And it can then use what it has learned to predict the activities of things it has never seen before. So we think that's really, really cool. So yeah, I think in this presentation, I told you that we can use these types of deep learning models to explore, sequence spaces that would be very difficult to access otherwise. In these types of protein engineering studies, many people oftentimes go, one mutation at a time. And so to go, and each round of doing that takes a very long time. So within one round to go, 30 to 50 mutations is, I think, pretty amazing. And it's enabled entirely by, the advances in deep learning. I showed you that we could collect a lot of data at high throughput using these cell-free assays. And then, it's still surprising and really interesting to me that, such a simple model, a linear regression essentially, can have really strong performance on learning the sequence function landscape and predicting the activity of other sequences. So I think sometimes you don't need, transformers and all these crazy things. A very simple model can be very powerful. And so, in the future, we're interested in things like we did activity and expression levels, but we're interested in more complex functions like maybe allostery or epistasis. It would be interesting to, even though I said it's, a simple model can be useful, maybe it would be interesting to look at more advanced models, especially when we want to do more complex functions such as allostery or epistasis. And then something we're interested in doing right now is what kinds of data are the most informative. So we have, data on combinatorial variants where you have, 30 plus mutations, but maybe it would be interesting to get single mutations or double mutations. And those are areas that we're actively exploring. So with that, thanks for listening. I want to acknowledge a couple people in the lab who are very helpful, including my PI, Tanya, Amy, who is a lab member who did a lot of the initial model building, and then Denise, who developed frame2seq in the lab, which we used to redesign our kinases. And if you're interested, the bioRxiv link is on this QR code, but I'd also be happy to share it if you are interested. So with that, yeah, thanks again. I'll just leave you with the conclusion slide. But if you have any further questions, I'd be happy to chat about anything that I talked about today.
Thank you very much, Dr. Kosuke, for this wonderful presentation. Mine is about the work that you did first about where you combined those three models where there was regression, and I think it is about the embedding variants that were used, where we see that one hot encoding seems to be giving better performance.
Yeah, I think it's this, right?
Yes, this one here. My first question is about why ridge regression is not like one would expect random forest to perform better, but what could be the reasons why you think ridge regression performed better in this context? But also you said the augmentation sort of parameter really contributed like nothing within the predictions. So what would be the way forward for better performances
Yeah.
in case like, yes, like should we get rid of it or there are better ways to improve it?
Yeah, I think those are really interesting questions. So first you asked about, the differences between ridge and lasso and random forest. Um, I don't know, to be honest. I think it is really interesting that ridge regression works so well, especially if you just look at something like ridge or lasso. They function very similarly, right? It's like different regularization strengths, but ridge consistently does significantly better. And I think if you look at many other papers in the field, we find that a lot of people find on many different types of data that ridge regression does very well. Um, and I think it would be really interesting to look at why it does so well. This is, a area that I'm interested in asking as well. So unfortunately, I don't have an amazing answer for you, but so far this is what we see. In terms of the augmentation, I think that's also interesting as well. I was not surprised to see that the augmentation didn't help when we used ESM embeddings, for example. And I think that's just because the ESM embeddings have so much information in them contained within them that's just learned by the protein language model. It probably already knows about, like evolutionary relationships, for example. And so that's why something like evolutionary density doesn't help. And protein language models have also been shown to know, have learned implicitly have learned structure just from these relationships. And so I think that's why things like delta delta G also don't appear to provide a big boost in performance. I was a little surprised to see that it didn't seem to provide a lot of improvement in performance in the one-hot encodings, I would say. And um, maybe I can speculate, I guess the speculation would be we picked residues that for example, were not highly conserved. And so maybe it might be that in that case, then maybe providing more information on evolutionary density isn't that important because already these residues are known to not be evolutionarily conserved. And so just the way we selected those residues might have contributed to that. And for similar reasons, it could just be that these residues don't really contribute to the stability of the protein. So it might just be that, just for our specific use case, the augmentation we chose didn't help. Yeah, that would be my hypothesis. I do see a
Yes.
question in the chat. Does your interpretable model's identification of functional important residues correspond to or help validate the established structural roles of the C spine and R spine? That's a fantastic question as well. I think it does. Let's see if I can pull up the slide that we are looking at. Yeah, I think it absolutely does help contribute to the things. It does correspond to things that are already known about these functional regions. So what's actually interesting is that so for example, I would say this T791 position shows up as the most important residue, and it turns out that that residue is, for example, conserved in other EPH family member kinases. And so we can see already consistency there within, like functional residues. And in terms of like the C spine and R spine, we do see that many of these residues are in very close proximity to those residues. I'm thinking, for example, this D706, which shows up as the second most important residue in our model, and this E684. These are very close to the C spine and R spine, and we find that they do have effects on the activity of the kinase. So I think it does corroborate some of the established roles of some of these functional regions. And in addition to that, there's things such as like the activation loop and the alpha C helix. We do find mutations around those areas as well. So I think we do corroborate known things about these types of enzymes.
Okay. I think for me, I'm satisfied with Dr. Kosuke's work, and really, it is a brand new sort of insight into understanding protein mutation and the kind of work that can be done in terms of applying machine learning, deep learning into predicting mutations. If I don't know whether there are other members in the audience with questions, but one question I can't leave without asking Dr. Kosuke is in terms of like for people who are trying to find a foot into this kind of work, maybe in a minute you could give us the starting point, because I know my colleagues here, some of them, these are fresh waters, the kind of things they could look out for. But also for people who are researchers in fields like mathematics and computer science, how they can write some good work within when it comes to protein structure, synthesis, and mutations.
Yeah. I think in terms of getting a foothold into this work, one thing that I'm more than happy to do, that, I think our lab and myself more than happy to do, is to share some of the data that we have, so that that people can explore it. And so we're still, we definitely want to do that and make the data publicly available. So, hopefully anyone can get, reproduce, for example, the ridge regression results that we did, or things like that. And so let me know if you're interested in that. I'm more than happy to share. I'll have to talk to my PI, but we've talked about it and we're more than happy to make that data available. And so, if people want to play around with the data, see what other things they can do, I think that would be I would be super interested in seeing what people can do with it. So I think that would be a really interesting way to get started. And I think generally I think there are other data sets out there that people have used to look at these types of, sequence function landscapes. And I think that just looking into, like the open data that's out there, I think is like a easy way to get at least started to learn like how to work with this type of data would be my suggestion. And I think a lot of you all are probably better statisticians or mathematicians than I am. I'm kind of a in-between engineer biologist person, so I really would be excited to see what other people think about these types of data.
That's wonderful, Doctor. And I think it's an interesting gesture and a great one. Really, data is a problem, and I'm sure we will have to reach out anytime from now.
Yeah.
I want to thank you again for saving up your time and talk to us and sharing this work with us. And I want to thank members who have turned up for this session. So I think we have reached the top of the hour and the end of the session. Thank you, Doctor, once again, and thank you, everyone. Yeah.
Yeah, of course. Thank you for inviting me. I enjoyed, answering your questions and chatting with you all. And again, if you're interested in, exploring some of the data that we have, just send me an email. I can reach out to my PI and we can see what we can do.
Okay.
Sure.
Thank
Fantastic.
you.
All right,
Yeah.
Doctor,
Thank
and
you so
to the
much.
members. All right.
Bye.
Goodbye, everyone.
Bye.
And
Have a
see
good
you in
evening for
the
you all.
good evening too.
Yeah. All right.
Okay.
Bye.